Let's begin our exploration of gene expression by examining prokaryotic transcription. We'll continue by looking at eukaryotic transcription and some of the big differences. In prokaryotic transcription, we need a single RNA polymerase in order to copy the gene from the DNA and transcribe it into messenger RNA. The initiation of mRNA synthesis doesn't require any primers or any of the other additional enzymes that were involved in DNA replication. It does, however, require a large holoenzyme. The holoenzyme is made up of two main components, the core enzyme, which is here, and then this sigma portion, which helps it actually clip onto the DNA. Prokaryotic transcription requires a promoter, which is a region that helps the prokaryotic RNA polymerase bind the gene and anchor itself in the correct orientation on the DNA in order to transcribe it. We also need a start site, which is the specific location that the RNA polymerase will begin polymerizing the new messenger RNA strand. And a termination site. This is where transcription will end and the RNA polymerase will fall off of the DNA. Transcription is made up of three phases: initiation, elongation, and termination. If we think of this all logically, it's pretty easy to understand. During the initiation phase, we need to get the RNA polymerase to bind to the promoter region on the DNA, get to the start site, and then in elongation, we start adding free nucleotides to the growing nucleotide chain. Once we get to the end, there'll be a termination sequence. This is where RNA polymerase is released and the RNA molecule is released so that it can go on to translation. The image here on the right might be a useful one to come back to as you try to understand each of these phases of transcription. The initiation phase occurs at the promoter region. The promoter region is a recognition and binding site for this large RNA polymerase molecule. It's upstream of the start site. Here we can see the start site is +1, that's just above 0. And we'll notice that there's a couple of promoter regions upstream from that at -10 and -35. That implies the upstream. And downstream is where the gene is located that we're interested in transcribing. You'll notice that these regions of promotion are ta-ta boxes, or TA-TA. You'll recall that T's and A's have 2 double bonds between them rather than 3 double bonds. This allows the RNA polymerase molecule to get in there a little bit more easily and begin to untwist the helix. So here we can see the helix being opened inside the enzyme. This happens about the -10 sequence. Now, you don't have to keep these -10 and -35 in mind, just for your own information if you like it. Once the RNA polymerase is bound to our unwound DNA, we have a transcription bubble. As transcription begins, we'll see that there's an mRNA molecule being synthesized in the transcription bubble and this unwound region of DNA. Now, we don't need gyrase or anything here because we're just opening up a very small region of the DNA in order to transcribe just the nucleotides inside that bubble on the template strand. So in short, initiation is about putting this large holoenzyme on the DNA and beginning the process of transcription by unwinding the helix. In the next phase, we have elongation. Elongation is simply adding nucleotides to this growing messenger RNA strand. The bacterial RNA polymerase also read the sense strand in only the 3' to 5' direction. So just like any other polymerase, they can only read in the 3' to 5' direction, which results in the new messenger RNA being manufactured in the 5' to 3' direction. We'll add in, just as we did in replication, nucleotide triphosphates. They come with their own energy to help the formation of the phosphodiester bonds between each of the nucleotides. Again, the transcription bubble, which has been enlarged here, contains our RNA polymerase molecule, which is the big green guy, the DNA template strand, and the growing messenger RNA transcript. After the transcription bubble passes, the now transcribed DNA is rewound and it leaves the bubble. Finally, we get to termination. The termination of transcription is marked by sequences that signal the polymerase to stop. These can be terminator sequences that are rich in uracils or perhaps a hairpin turn. A hairpin turn is demonstrated here. As the polymerase moves along this strand, it runs into a hairpin turn and literally falls off the DNA template. Once this happens, the DNA and RNA hybrid within the transcription bubble dissociates, so the messenger RNA strand is released. The RNA polymerase will then release the DNA, and the DNA will rewind. So now we have everything dissociated. The messenger RNA is now ready to go on and be translated, and the DNA has become freed from the RNA polymerase, and the RNA polymerase is ready to go on and transcribe another gene elsewhere. Now, in prokaryotes, transcription is coupled to translation. Because the transcription occurs out in the cytosol, there is no nucleus. Ribosomes are right there and able to begin translation at the same time. So what you can see here are strands of messenger RNA that are attached to the original DNA molecule, and they have ribosomes on them, translating them at the same time as the RNA is being transcribed. Also in prokaryotes, we'll see grouping of functionally related genes. These genes might all be involved in one pathway, and they can be regulated together. Thus, they are called an operon. So the RNA polymerase will transcribe these genes in sequence, and then they'll each be made individually as a protein. Let's watch a quick animation to help us understand how bacterial transcription works.
In bacterial cells, the transcription of a gene or group of adjacent genes called an operon is controlled by a set of proteins interacting with specific sequences in the DNA molecule, leading to the initiation of transcription and the termination of a transcript. Transcription is initiated just beyond the yellow promoter region and is terminated at the green terminator site. The sequence of the promoter at positions 35 and 10 nucleotides upstream from the transcription start site are critical to initiating transcription. In order to initiate transcription in bacteria, core RNA polymerase and sigma factor form a holoenzyme. Sigma factor recognizes the sequence of DNA at the promoter region. This holoenzyme then binds to the promoter region of the DNA molecule, forming the closed complex between RNA polymerase and the DNA molecule. By breaking hydrogen bonds between bases in the double helix, the closed complex is converted into the open complex. The formation of the RNA transcript from nucleotide triphosphates then takes place as RNA polymerase is forming the new transcript, starting at the 5' end of the transcript. New nucleotides are added onto the free 3' end. The initiation of transcription is completed when sigma factor is released. As elongation of the transcript then continues, the RNA polymerase moves down the DNA molecule and the next sequence in the chain is opened up. As RNA polymerase moves down the DNA strand, the DNA molecule entering RNA polymerase unwinds, forming the open complex, while the DNA exiting RNA polymerase rewinds to form double helix. Ribonucleotide triphosphates are added to the chain, and the growing RNA transcript continues to elongate as new DNA sequence enters the open complex region. The new RNA transcript initially forms an RNA-DNA hybrid complementary to the template strand, corresponding to the sequence of the coding DNA strand. In this way, the DNA double helix is opened, transcribed, and reclosed with minimal stress on the DNA molecule. One mechanism of bacterial transcriptional termination involves the binding of the Rho protein to the RNA. It is a helicase that moves in the 5' to 3' direction toward the RNA polymerase. Between the Rho recognition site and the terminator site, a stem loop structure is formed by the sequence in this region. This stem loop structure halts the movement of RNA polymerase, and the Rho protein then moves to the RNA polymerase complex, causing the complex to dissociate and terminating transcription, resulting in the full-length mRNA transcribed from the gene.
The basic mechanisms of transcription by RNA polymerase is the same, whether it's eukaryotic or prokaryotic transcription. So in this section, as we consider eukaryotic transcription, we'll look at how eukaryotic transcription differs from prokaryotic system. First of all, eukaryotic transcription involves 3 different RNA polymerases. Now don't worry, only one of them at a time. RNA polymerase 1 is the polymerase that's involved in making ribosomal RNA, so we're not considering that now. RNA polymerase 2 is the one we care about. This is the one that's involving the transcription of messenger RNA and also some small nuclear RNA that we'll see shortly. RNA polymerase 3 is the polymerase that's involved in transcribing tRNAs and some other small RNAs that we're not really concerned about right now. Each RNA polymerase recognizes its own specific promoter and thus only transcribes the RNA that it's supposed to. The initiation of transcription in eukaryotes differs a little bit. It requires a series of transcription factors. These are essentially like escorts. They're necessary to get the RNA polymerase II enzyme to the promoter and initiate the gene expression. Basically, it's a more complex mechanism and involves many other transcription factors to help this big old RNA polymerase latch on to the promoter region and initiate gene expression. It basically works like this. The transcription factor recognizes and binds to the TATA box, which is the promoter sequence. And then the other transcription factors are recruited to anchor it on there properly, and the initiation complex begins to build. Ultimately, the RNA polymerase associates with all of these transcription factors and then the DNA, forming this whole initiation complex. From here, transcription will begin. And although termination sites exist, they are not that well defined in eukaryotes as they are in prokaryotes. So other than these couple of differences, everything goes along in just the same way. Once the initiation complex is formed, polymerase is able to polymerize the growth of a new messenger RNA strand, and that messenger RNA strand drops out as they reach the termination sequence. But then, what happens to the messenger RNA? In prokaryotes, we've already recognized they have no nucleus, and so transcription and translation occur concurrently. As the messenger RNA is being transcribed, ribosomes jump on it and start assembling the polypeptide chain in the process of translation. There is no processing necessary, no modification necessary to the messenger RNA. In this case, prokaryotes can only regulate how fast and how often genes are expressed at the level of transcription, because once it's transcribed, it is expressed. However, in eukaryotes, things are a little bit more complex. Eukaryotes have a nucleus. The DNA is located in the nucleus, and thus transcription occurs in the nucleus to form a molecule that's called pre-messenger RNA. The genetic organization of eukaryotic DNA is much more complex also, and so it's necessary to process or modify this pre-messenger mRNA in order to make it mature messenger RNA. The mature messenger RNA is the RNA that will leave the nucleus and go out into the cytosol to be translated by ribosomes that are either free-floating or are on the rough endoplasmic reticulum. So let's look in more detail about what happens when eukaryotic messenger RNA is processed. We have 3 main events: addition of a 5' cap, addition of a 3' poly A tail, and removal of the non-coding sequences, or introns. We call this pre-messenger RNA splicing, and it's done by something called a spliceosome that we'll look into here shortly. When we add the 5' GTP cap, it's a guanosine triphosphate. This is going to protect the DNA from degradation. Other enzymes are cruising around, waiting to gobble it up and degrade it. Without the cap, it might get gobbled up before it's even translated. The addition of a 3' poly A tail simply adds a lot of adenine residues to the end of the messenger RNA. Again, this protects it from enzymes that might be chasing its tail and trying to degrade it. So both of these 5' cap and 3' poly A tail protect the messenger RNA molecule. The third part is a little bit more complex. We're going to remove these non-coding sequences. Interspersed throughout the messenger RNA are regions of non-coding material, or inactive material, that we call introns. You can see here the DNA template has the code for these introns also. The introns are removed in the process of RNA splicing, and then we have a mature messenger RNA with a 5' cap and a 3' poly A tail. In this figure below, you can see the portions that are removed here in purple. Our mature messenger RNA is simply this yellow region. And these outloopings are going to be introns that will be removed. So introns are inactive or non-coding sequences. Exons are expressed. These are the live sequences that will be translated into the final polypeptide chain. Let's look a little closer at how that works. When we need to remove an intron, we'll involve spliceosomes. Spliceosomes are composed of protein and small nuclear RNA. We call these complexes small ribonucleoprotein particles, or SNRPs for short. They have RNA sequences in them that recognize specifically the end of a particular intron. They'll bind with this end of the particular intron and cluster to form a spliceosome. Once in this position, the 5' end of the intron is removed and forms a bond at what's called a branch site. And then the 3' end of the intron is cut. And we basically end up with a little loop here that's called a lariat loop. And exon 1 and exon 2 are glued back together. These messenger RNA modifications associated with pre-mRNA processing are coupled with transcription. That is, they happen at the same time as transcription. So as the new pre-mRNA molecule is created, the introns are cut out and the 5' cap is put on. And as soon as it's finished, we add the 3' poly A tail. A single primary transcript could be spliced into different messenger RNAs by the inclusion of different sets of introns and exons. You may recall when we first examined the human genome, we found out there were only about 25,000 to 35,000 genes, not really many more than a fruit fly, and we were quite shocked. But now, as we've learned more about epigenetics and such, we've recognized that each of these genes can encode for more than 80,000 different messenger RNAs, and thus 80,000 different polypeptide chains. So this alternative splicing, as we call it, allows us to use one gene to create possibly several different polypeptides. So this is a place, again, where we're expanding beyond the one gene, one polypeptide concept. We are in fact learning that one gene could code for multiple different polypeptide chains, depending on which introns are cut out and which introns may be left behind as coding sequence in a different protein. 15% of human genetic disorders are due to these altered splicing, accidents in splicing. So this could be another way that we obtain some genetic variation. Also, we've recently noticed that 35 to 59%, let's say an average of 40% of human genes exhibit some form of alternative splicing. That is, they code for different proteins. This is fascinating, great new information. This animation to help us better understand messenger RNA splicing.
Eukaryotic genes consist of coding sequences of nucleotides called exons and non-coding sequences called introns. The product of transcription, called the primary transcript, is formed with a 5' cap and a 3' poly A tail. The intron sequences are then removed by the process of RNA splicing. The intron-exon junctions are recognized by small nuclear ribonucleoproteins called snRNPs. These SNRPs then cluster together, forming a larger complex called a spliciosome, which carries out the removal of the intron. Splicing involves cleavage of the 5' end of the intron, forming a looped structure called a lariat, joining the two exons together and releasing the intron. One advantage of the splicing process is that a single primary transcript can be spliced into different messenger RNAs by the inclusion of different sets of exons. This process, called alternative splicing, explains how the 30,000 genes in the human genome are able to encode the 120,000 different translated messenger RNAs reported to exist in human cells. On a larger scale, it has been proposed that an advantage of gene splicing may be that during the course of evolutionary history, various combinations of exons may have been created by exon loss, duplication, and reshuffling. Changes in the sequence of the primary transcript could be due to mutations in the DNA of the gene or to mutations in an intron excision mechanism.
So now let's take a quick card quiz to finish off this section. What is the function of the 5' cap and the 3' poly A tail? snRNPs, or SNRPs, are responsible for splicing out the non-coding exon regions. Where does the transcription take place? RNA polymerase would read from 3' ATCTTA through the 5' end and make which of the following RNAs? Here are the answers to your card quiz. If you got all of those correct, you're well on your way to a good understanding. Now would be a great time to spend some time diagramming the process of RNA transcription. What happens during initiation? What happens during elongation? And what happens during termination? Perhaps this simple overview image is a good guide for you to consider as you try to put together your diagram.